Good morning, guys. It's Wednesday.
Morning.
Good morning. Wednesday, October 28th. So I assume that you can hear me since you responded already. So we have one more lecture on chapter 8, and then we'll have a test on Friday. So I probably already mentioned to you that we're going to go back to 30 questions, right? So and so the exam will, as far as the difficulty, the exam is similar to the first test. So I do suggest that you guys check your equipment prior to the test. Make sure it works. Make sure that some of you had some technical issues last time. Make sure you use Google Chrome, right? So that runs best with the what do you call it? Canvas. And as far as the figures, so a few of you told me that you had troubles seeing the figures. So what I'll do is actually maybe I'll reopen those practice problems with some figures in them that I did for you last time. And you just open it again, make sure you can see the figures, okay? Make sure you can see the figures. So obviously if you see the figures on that, you will see the figures on the actual test. All right, so we have quite a bit of material to cover today. I have to go through 28 slides, so we better get going. So we talked about double-stranded DNA last time. We talked about aDNA, bDNA, zDNA, and so here's our two strands. A pairs with T, G pairs with C in DNA. So obviously the two chains differ in sequence. And specifically, how do they differ in sequence? They are complementary, right? So whatever you see C on one side, you'll see G. And the second important observation is the two chains run anti-parallel, right? So if this one runs 5 to 3 prime, 5 prime to 3 prime in this way, this will be running 5 prime to 3 prime in this way. So yeah, so this immediately suggests a possible copying mechanism for the genetic material, right? So if you use the left strand as the template, then basically you will create the copy of the right hand strand, right? And if you use the right hand strand, you will create a copy of the left hand. And so the two mother strands will give you two daughter strands, and from two you go to four strands, which will be each of which will be double-stranded. So it's a pretty simple and elegant way to replicate. So here is a illustration of that, right? So obviously the DNA, when it's double-stranded, cannot replicate. The two strands have to separate, right? So there are lots of enzymes which participate in DNA replication. They pull the two strands apart. And you can see here, so the new strand will start to be synthesized from the 5' and go to the 3' direction, right? So so the new strand is shown here. It's a daughter strand, and it's being synthesized downward. Here on the right-hand side, the new strand is coming from the bottom and is immediately undergoing formation of the double helix with the parent strand, and it's undergoing synthesis from the 5' to the 3' direction. So this is how this is made, and this is how this is made. So so once the DNA replication is complete, you immediately have two double-stranded pieces of DNA. Again, the enzyme that puts it together is DNA polymerase, right? DNA polymerase. And if you make messenger RNA, that will be RNA polymerase, right? So the difference will be is obviously the type of nucleotides which are being used, right? So DNA polymerase will use deoxynucleotides, and DNA RNA polymerase will use just nucleotides with the hydroxy group at position 2'. Okay, so that's replication. So as far as RNA, a couple things we need to mention about RNA. So we know it's a code carrier, right? So DNA stores the code, and messenger RNA will carry the code. Will carry the code to the ribosome to make proteins. So it's made from DNA template, and in general, it's a single strand. So RNAs do not form double-stranded sequences as easily as DNA does. So it has ribose, contains uracil instead of thymine. We know that. And also one messenger RNA may code for more than one protein. So we'll see that in a moment. And so together with transfer RNA, transfer genetic information from DNA to proteins. So here is the space filling representation, right, of messenger RNA. So you can see it's single-stranded. It's also it also has base stacking, just like we talked about. Okay, and so it's a right-handed stacking pattern of single-stranded RNA. So the bases here, you can see the bases are in yellow. You can see they stack upon each other, right? The bases stack upon each other. Phosphorus in orange. Now I'm sort of colorblind, so I don't really see the orange, but it has to be there somewhere. And the riboses and the riboses are in green. Is that orange right there?
No, we can see the orange though. It's like.
Is that there, right there?
Yes, yes, it's really small.
Mm-hmm. Okay, and the phosphate oxygens are in green. All right. Yeah, so if the messenger RNA, if it codes for one gene only, it's called as monocystronic, and if it codes for a few genes, like 3 genes here, it's polycystronic, right? So messenger RNAs differ in their length and the number of genes they can encode for. All right, so another peculiar thing about nucleic acids is these palindromes. So you know there are palindromes are words and phrases which are the same when read backward and forward, right? So civic, read left to right or right to left, same word. Race cars, same word. Rotator, there's a long one. The Finnish word, I guess it's a very long palindrome. And there is even Greek, ancient Greek, wash the sin as well as the face. So with the nucleic acids, so these will be sort of inverted repeats, right? So for example, you see here, we will, so let's say you have one strand that has a particular sequence, and then the complementary strand that sort of where another sequence starts, where this sequence ends, basically will be the inverted version of that. The inverted version of that, right? Which means is that the complementary portion of this highlighted one will be complementary to the original one, right? So what this does, this gives rise to the ability of the RNA to form double-stranded portions with itself. So in other words, a single strand will be creating loops, which will come back and become complementary to an earlier version of the sequence of nucleotides. So you can already kind of see that, right? So let's say if you bend this, so G will pair with C, T with A, C with G, C with G, and so on and so forth. So these will be mirror repeats. So these are not as important in folding, but these kind of palindromic sequences are, and they form what's known as hairpin structures. So you would have this inverted repeat, right? So a sequence here, and here it's inverted repeat on the same strand. And there is some kind of sequence in between. It doesn't really matter what it is. What matters is that it's long enough, let's say it's 4 nucleotides. What matters is can accommodate for the loop so that one sequence, let's say, goes this way. So it's a 5' direction. And then loops around, and then the inverted repeat forms a complementary base paired region, right? So these are pretty common in RNA. So these palindromes or inverted repeats, more correctly, and specifically they play a huge role in organizing messenger RNA structure. So if DNA is all double-stranded, RNA is single-stranded, but it's full of these inverted repeats. So
could you go back to that slide real quick?
Yeah.
So for hairpin, is it always this sequence T A G C G T, and then the inverse on the other side? Is it always this? Is this like the code for it? Is there different ones?
Or different ones. So as long as it's complementary, that's all that matters.
Okay. So if we're like given a strand like the one above the red, we just would recognize that, okay, there's 1, 2, 3, 4, 5, 6, and then 4 that are different, and then the other 6 are complementary to the first.
Right. Mm-hmm.
Okay.
So are hairpins like a type of mirror, or are they like totally different because they have that middle piece that isn't the same?
Yeah, so this will obviously, well, it's not really a mirror because these are complementary, right? These are not identical.
Okay.
So,
oh, okay, I see.
Yeah. Yeah, you just have to have a complementary sequence some distance apart from the original sequence, and then it will be able to form a hairpin. So you can see here, so this is, let's see what this is. This is RNA component of enzyme RNase-P. So RNase-P, I think it's an enzyme that participates in synthesizing the ribosome. No, take it back. Contains a protein component, functions in the processing of transfer RNA. Take it back. So processing of transfer RNA. So so you can see there are actually a whole bunch of these hairpin loops, right? So again, so this sequence and that sequence, they're complementary. And another important thing is that obviously now we have we can't, with RNA, we can't have Watson-Crick pairing because there is no thymine. So uracil has to be used for complementary base pairing. And one of the common ones is actually guanine and uracil. Guanine and uracil is a very common base pair in RNA. So let's see if we can find it. GC, GC, GC.
It's down here.
You found
on the
one?
left. Yeah. Here, I'll circle it with annotate.
Yeah, right
Yeah,
here.
yeah,
Connected by dots. Yeah.
Yeah. So they occur once in a while, and that's actually very characteristic of messenger RNA. Oh, there's another one. So you can see here, to remember this, so uracil, so here with guanine, so you will not, so basically you would utilize one hydrogen bond acceptor, one hydrogen bond donor. In uracil is the other way around, right? Is the other way around. And there's this NH2 that is kind of not really participating, And so, one thing is something I need to mention about base pairing, because for a long time people thought that these hydrogen bonding properties of base pairs, they were the main driving factor behind the formation of the double helix. Whereas now it's actually believed that that the complementary hydrogen bonding plays a role to make the double-stranded or double-stranded portions more stable. But it turns out is actually it's geometrical preferences of these bases which make these double-stranded portions more stable. And once they click into place, then the hydrogen bonds form. So in other words, hydrogen bonds assist, but they're not the primary recognition factors, right? And so they play a role, but not the major role. And that has been done. There is a scientist at Stanford University, Eric Kuhl, who's done a lot of work in this area. And basically he synthesized a lot of these nitrogenous bases, synthetic nitrogenous bases, and he would remove, let's say, instead of this oxygen here, he would put a fluorine here or something, or methyl group here, that would be incapable of forming a hydrogen bond, but the DNA will be just as stable as it was with the hydrogen bond. So where he basically with his research, he showed, I think it's a K actually. I think with his research, he showed that hydrogen bonds are not the primary driving force, and you can make highly complementary, highly stable, double-stranded segments of DNA that lack hydrogen bonding capabilities. All right, and then, for example, the pair GU shows us, right? So basically, it's not a very strong base pair because there is only 2 hydrogen bonds, and yet it's quite prevalent in RNA structure. Now, and there is also another type of hydrogen bonding I want you to be aware of. There is what's known as a Hookstein base pairing, which is also referred to as non-canonical base pairing. Specifically, if you look at this, so here you have canonical Watson-Crick, right? Watson-Crick, where we have G and C. But then once you form G and C, you still have oxygen here with a lone pair, and you still have a nitrogen position 7 with a lone pair. And so this oxygen, right, position 6, and nitrogen position 7, they're both hydrogen bond acceptors. So they can form hydrogen bonds with a different nitrogenous base coming this way, right? And so this will, for example, this particular one is N-methyl 7-methyl guanine, and you can see there is 2 hydrogen bonds possible here, and there's even phosphate that can participate in that. So this is particular motif where this nitrogen lacks hydrogens, the 2 methyl groups, and so so adenine will form a base pair with this kind of dimethyl guanine and a different kind. So so these kind of hydrogen bonds are capable of forming different types of DNA structures. So for example, we talk about double-stranded DNAs, but there are, you can have triple-stranded, you can have tetra. So, or they call the tetramers. So where you have 4 strands running together, right? And quite common. So for example, something like this: base, 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 and then there's phosphate backbone, ribophosphate backbone. Can be parallel, can be anti-parallel, but this will be a tetramer. Tetramer. Where you have 4 strands coming together and forming a higher structure. Have you guys heard of the term? Have you heard of it? Go ahead.
Can you explain what the Hoogsteen is again?
Hoogsteen is a non-canonical base pairing, specifically involving the nitrogen 7 and oxygen 6. Because both of these are hydrogen bond acceptors, so they can form hydrogen bond with hydrogen bond donors. Have you guys heard of telomeres?
Yes.
What are they?
They're regions at the end of the chromosome that don't carry much genetic information, but they get shorter each cycle of replication.
Yeah. So thank you. So telomeres are primarily tetrameric DNA, and so they actually formed from this kind of arrangement where you have 4 strands running together, and so the enzyme actually that synthesizes them is telomerase, right? Telomerase can put these together to 4 strands together, and as you said, that telomeres can get shorter and shorter, and that's could be one mechanism in aging, for example, that our DNA gets shorter with every replication. So the more times the cell divides, the more DNA is lost. And cancer cells actually express these enzymes telomerase, which will replenish these telomeres, right? Because if cancer cells divide so much more frequently, you would think they should run out of telomeres very quickly, but that's part of the problem. Part of the problem is they overexpress this enzyme telomerase, which is important in childhood when there's a lot of cell division, right? When the person grows, there's a lot of cell division, tissue differentiation, lots of telomeres getting shorter and shorter. And so telomerase, the enzyme, is overexpressed at a young age. But then once we get older, telomerase becomes inactive, but cancer cells find a mechanism to keep the telomerase active, and they continue elongating telomeres so that the cells will not, so the telomeres will not get too short and the cell will go into apoptosis. All
So
right.
does the Hodgkin require the N7 and O6 because adenine doesn't have an N7 and O6, or would it only happen in guanines?
So so Hookstein base pairing. So you're asking what other bases can participate?
Yeah, because you said it usually, like, does it usually involve the N7 and N6, or does it have to have?
Yeah, so it's N7. So this is a hydrogen bond donor. Wherever you see it's involved
Okay.
in hydrogen bonding, that's a sign that it's a Hookstein type base pairing.
Okay. Okay.
And it's usually, you see it's on the side. Usually Hookstein is actually an addition to the canonical base pairing, Watson-Crick. So this is known as Watson-Crick base pairing, and this is Hookstein base pairing.
Okay, perfect. Thank you.
All right, so DNA can undergo denaturation. So just like with proteins, when you heat them, when you heat proteins, they will lose their three-dimensional structure, they will denature. Same thing with DNA, right? So if you heat DNA, the covalent bonds will remain intact. Genetic code will not be unless you overheat it, obviously, over 100 degrees Celsius. But the genetic code will remain intact, but hydrogen bonds will break, and the two strands will separate, right? So this is process of denaturation, and if you go back, it's annealing. So the two strands will come together and form double-stranded DNA. And you can, if you continue a denaturation process, then you will completely separate the two strands from each other. So denaturation can be induced by high temperature or change in pH. So it can be reversible, and the reversible process is called annealing. So now this process forms a basis for a polymerase chain reaction. So we're gonna look at that in a moment. There are 3 slides on polymerase chain reaction. I sort of uploaded them in a revised version of these slides to Canvas. So you can look at those 3 slides. If you want to keep the original slides, you just have to copy and paste those 3 slides into the original document, or you can just adopt a new document. It's up to you. And so DNA denaturation is commonly monitored by UV spectrophotometry. And so this is actually a very interesting phenomenon. When you have base stacking, when you have aromatic systems stack upon one another, and you excite them with light, oftentimes this will be quenched. And it's quenched because, so let's say this pi system goes to pi star, right? But then an electron will be transferred from here to there, which will quench this pi star. Okay, so this other pi system, when it's stacked upon the another one, they can quench each other, and so the absorbance will be lower because of that. Okay, so when you separate, when you break the base stacking, then the absorbance increases significantly, and you can see here. So this is what's known as a melting temperature. So melting temperature is the temperature at which 50% of denaturation happens, right? So there's a blue, there's a red. So which DNA fragment is more stable, or does not denature as easily?
Blue.
The red one.
Uh, blue or red?
Oh, blue.
So blue is more stable.
Yes, because it requires this temperature.
Are you sure?
The red one is more stable.
Red one is more stable. The red one requires higher temperatures, right? So so you can see to denature half of all the DNA, you need 85 degrees Celsius. To denature half of the DNA here, you need only 83 degrees Celsius, right?
So TM equals the percent of DNA that's been denatured.
TM
50%.
is the temperature at which 50% of DNA is denatured.
Okay.
So so different fragments of DNA will undergo different kind of will have different TMs. Now let me ask you something. So two fragments of DNA, one is rich in GC base pairs, one is rich in TU base pair. In AT base pairs. Which one will have a high TM?
GC,
GC, right? And why?
because they're stronger because it's 3 bonds rather than 2.
Right. So DNA fragments which are richer in GC base pairs will be more stable because there are 3 hydrogen bonds holding the 2 strands together, as opposed to 2 hydrogen bonds holding the AT together, right? So it's pretty straightforward to actually process to predict this TM, right? You have a DNA fragment, you know the sequence, and you can predict the TM, whether it's going to be high or low. All right, base composition, yeah, GC increases TM. Also DNA length, obviously. The longer the DNA, then harder it is to separate the strands. pH and ionic strength, salt increases TM. So as far as salt, I think because the ionic strength, because DNA is actually salt by itself, right? So it's a multi-ionic salt. It's a polyphosphate. And so obviously if you have high ionic strength, it will keep this polyphosphate together. It will not allow it to sort of dissolve into solution, right? It's gonna work against the ionic gradient. And so the ionic strength will kind of pressurize the DNA and will keep it together. At least that's my explanation. And so here you can see this is the GC content, right? As the GC content increases, the melting temperature increases as well. And you can see here, for example, when you have 30% GC content, TM is 80 degrees Celsius. When you have 70%, it's over 100. And this is electron micrograph. Picture of it, you can see here, some of it is easy to see, some of it is not. This is double-stranded, but this is still stays double-stranded, probably high GC content. So these ones, and apparently these ones are AT rich regions, and you can see now the 2 strands are falling apart, are coming off each other. So you kind of have these loops, right? Loops where the two DNA strands separate. And so it's been shown that these ones AT rich and these ones that do not come off each other, they are GC rich. All right, so let's briefly talk about polymerase chain reaction. So, so it's based on the property of DNA to undergo denaturation and annealing. Was the person who discovered it, Mullis. Professor Mullis, he was awarded Nobel Prize for the discovery of polymerase chain reaction, which totally revolutionized many areas. For example, identification of various species of extinct creatures through fossils, right? DNA preserved in fossils. And oftentimes you just need just one molecule of DNA to actually to synthesize many copies of that DNA. For example, from one molecule of DNA, within hours, you can actually synthesize 1 million copies of that DNA. If you synthesize 1 million copy of that DNA, you can easily do the sequencing, and you can actually sequence the entire DNA, and obviously then you can compare the species. Also in forensic medicine, right? So if there's some DNA left at the crime scene, so no matter how little DNA is left, you can still amplify that with polymerase chain reaction, obtain enough of the DNA for sequencing, and you can, that way you can actually identify a particular individual who left his DNA at the crime scene. So, so can amplify regions of interest and plasmids as well. So plasmids, you know that they're show circular DNA. So we're not talking about those in this class. It's more from the biology, but you can also amplify those. So, so what do you need? You need target DNA, right? So even though it's maybe one molecule, but you need that. You need primers. You need primers, which will be complementary to the target. You need nucleotides. So for the DNA synthesis, you need deoxynucleotides, right? So deoxyadenosine triphosphate, cytidine triphosphate, guanosine triphosphate, and thymidine triphosphate. So these are deoxynucleoside triphosphates. And you need thermostable DNA polymerase. And we'll talk about that in a moment. It has to be thermostable. Remember, the enzymes can be denatured easily. So if we're going to heat this reaction mixture, the enzyme has to be thermostable. So you place the mixture into the reactor, melt DNA, cool, and so let's look at a figure is worth 1,000 words. So this is how it's going to work. So let's say this is the piece of the double-stranded DNA, right? The 2 strands. And this is the region in dark blue. This is the region that's going to be amplified. So what we're going to do, we're going to heat to separate the strands. So the DNA will denature. And then we're going to add synthetic DNA oligonucleotide primers and cool the reaction mixture. So usually to separate the strands, we do it at about 100 degrees Celsius, but then you cool it down once you add the primers, you will cool it down to about 60 degrees Celsius, and the primers will anneal. So the primers will actually form complementary base pairs at the beginning, at the basically 3, 5 at the 3' ends of the DNA to be amplified. And remember, the new DNA will be synthesized in the 5' to 3' direction, right, which is opposite of the template strand. So we add the primers. Now we're going to add this thermostable Taq DNA polymerase. Taq basically stands for, it's a T aquaticus. It's an organism that lives near thermal vents. And so it, you know, there are organisms that live at very high temperature, and they have developed enzymes which are stable at high temperatures. And so for the DNA polymerase, you cannot really use a human one because it will undergo denaturation. But you can use the DNA polymerase from this thermostable organism. All right, so DNA will be synthesized starting from the primer, 5' to 3' direction. And so this piece will be, this DNA will be amplified. But then the same thing will happen here. The same thing will happen here. Starting from 5', you're gonna make this, you're gonna go in the 3' direction and copy this one. And then again, you repeat, so it's very simple. Then you repeat steps 1 and 2 again. So you separate heat to separate the strands. Right now you have four strands, and you add the primers. So the primers will form complementary base pairs with each of them. And then you repeat the DNA synthesis, step 3, you add the DNA polymerase. And now you have actually 8 fragments of DNA. 8 fragments of DNA. Continue on. 8, repeat steps 1 through 3. 8 becomes 16, and so on and so forth. So after 20 cycles, the target sequence has been amplified about 1,000,000-fold, right? And this can be done within hours in a thermocycler, so in this apparatus which performs PCR reactions. So this is all automated. A person no longer does this in the lab. It's just done by specific equipment that you can buy. Okay. All right, so that's polymerase chain reaction. So the last topic I want you to chat with you about is some chemistry of DNA. So DNA can undergo mutagenesis. We know it's a bad thing, right? DNA undergoes mutations, which can cause some really bad health effects, and cancer is one of them. And so deamination is one of them. So cytosine, for example, can undergo deamination and it becomes uracil. And methylcytosine can become thymine. Now, the first reaction, cytosine to uracil, so these even deamination reactions are quite common, right? And so the first reaction is extremely important. And that's probably the reason why DNA does not have uracil. DNA does not have uracil. Instead, DNA has thymine. If DNA had uracil, imagine each deamination process of cytosine, for example, would give you uracil. And then you would never know if this uracil was there to begin with, or it's uracil which is derived from cytosine that underwent deamination, unwanted deamination. And so DNA repair mechanisms, DNA repair enzymes will not be able to tell apart which uracil is which. And that will lead to a huge number of DNA mutations, and eventually the genetic code will not work because it will be compromised. But when your DNA has a thymine, so by adding this 5-methyl group, you know that DNA should not have uracil. It should have thymine. So whenever uracil is found in DNA, the DNA repair enzymes already know that this uracil came from cytosine. And so what needs to be done? This uracil needs to be excised, it needs to be taken out, and cytosine will have to come in, right?
Can you explain that one more time?
Explain one more time. Which part? So the primary reason why DNA does not have uracil and has thymine is because the deamination reaction would also give you uracil, right? And then you would never be able to tell which uracil was there and is part of the genetic code, and which uracil should not be there and arose primarily through the deamination, spontaneous deamination reaction. And then genetic code will not work anymore because it's all going to be messed up. But if you have thymine instead of uracil, you know that if there is uracil in DNA, that's clearly not part of the genetic code. The only way uracil can find its way into DNA is through deamination of cytosine.
Okay, I see now. So these are all, so cytosine, when it's deaminated, turns into uracil, 5-methylcytosine turns into thymine. So these are all constant.
Yeah, but these ones, this is very rare, Right.
Very rare. Okay, so cytosine to uracil, that's more common.
Yeah, cytosine to uracil. Yeah. Hmm.
But because it's more easily, I guess, recognized by the mechanisms in DNA, it's easier to convert back into thymine because uracil isn't found in DNA. Is that what you were saying?
Well, you don't convert uracil into thymine. You convert uracil into cytosine.
You know, that's what I meant.
Yeah. So
Yeah.
DNA repair enzymes, every time they see uracil, they know instead of uracil, there should have been cytosine.
Okay.
And they will take uracil out and put cytosine in. Okay, so there are a couple other deaminations. This is the one I was telling you about, hypoxanthine, which is the precursor for the nucleoside, what's the name? Inosine. Inosine is a nucleoside of hypoxanthine. Comes from adenine. So there's a bunch of deaminations. So these primary amino groups can undergo removal, and you have the carbonyl instead of them. So so very slow reactions, but the net effect is significant. About 100 cytosines undergo deamination per day in mammalian cells. But there are DNA repair enzymes, fortunately, that come in and fix that for us. So depurination. So we talked about why purines are better in terms of leaving, right? So they have lots of nitrogens, they can be protonated more easily, and they can come off more easily, right? So depurination is a very important process in human DNA. So you form nitrogenous base and the apurinic residue. So apurinic means without purine. So you can have the free hemiacetal here, no nitrogen anymore. And significant for purines, about 10,000 lost per day in mammalian cell. Oxidative damage, another important damage in damage process in DNA. Obviously, reactive oxygen species, we know that, right? So we know that reactive oxygen species can be quite damaging. Chemical alkylation, methylation of guanine. So the position N7 on guanine I was telling you about, right? I told you about that. Can undergo alkylation. So it's also DNA damage. But cells have mechanisms to correct most of these modifications. And if these mechanisms don't work very well, the correction mechanisms, then that can lead to things like cancer, right? So, so DNA damage is a normal process as long as it's kept under control through DNA repair mechanisms, then it's fine. And in fact, DNA damage plays actually a huge role in evolution. Why do you think that is?
Diversity.
Diversity. You have to have DNA damage. If there was no DNA damage, we would not evolve, right? So we will not be able to adjust to the environment. So the evolutionary process incorporates DNA mutation as a big part of it. So as long as it's kept at bay, under control, so that there are some mutations, but not overwhelming, that can lead to the death of the organism, but rather adaptation, rather the ability to adapt to new environmental conditions through inheritance of the more beneficial genes, mutated genes, then DNA mutation is actually a highly favorable process. So here are some alkylating agents. So the most common one in biological system is this S-adenosylmethionine. It's abbreviated as SAM. Whenever you see SAM, you know it's a methylating agent, biological methylating agent, and it's an SN2 reaction. Nucleophile attacks the methyl group, and this leaves, right? Then a bunch of other very strong methylating agents. So for example, these nitrosamines, very strong carcinogenic species. Dimethyl sulfate, also SN2 reaction, right? I told you DNA is nucleophilic. So all these nitrogens have lone pairs. So nitrogen mustard. So actually many, for example, dimethyl sulfate or nitrogen mustard, they use as cancer agents, That's interesting. They used as anti-cancer agents. So they kill cancer cells by damaging DNA in cancer cells. All right, radiation, ion. So basically UV light, we talked about that. There was one particular process with UV light I wanted to show you. So obviously ionizing radiation, X-rays and gamma rays, right? So X-rays are fine if there are not so many of them. If there are too many of them, that can lead to DNA mutations as well. The one that I want to show you is called pyrimidine dimers. It's a process induced by UV light. So for example, let me see if I can quickly draw this. So let's say I have one pyrimidine. Usually it's thymine, but okay, let me just do this on example of uracil, even though it's not DNA relevant, but it will give you an idea what this dimer is. So this undergoes, it's like Diels-Alder reaction, only it's 2 + 2. Remember, Diels-Alder is 4 + 2, and this will be 2 + 2. It's a 2 + 2 cycloaddition, where these electrons come this way, and these electrons come this way, and you will form a cyclobutane. Cyclobutane like this. So it's a covalent bond formed within DNA, which causes DNA damage, and it's irreversible. These cyclobutanes don't break up easily. And when that happens, obviously the genetic information is compromised. Not only that, it also changes the shape of the DNA. So you can see here. So the dimer is actually shown here. You can see the cyclobutane right there. Yes. So there are two pyrimidines. These are thymines, as I told you. These are thymines. So instead of uracils, obviously uracils are not part of DNA. So we add these methyl groups here, and there'll be methyl groups there. So and you can see once this happens, right? So the stacking is interrupted, and this introduces kind of a kink in the DNA. And so the not only that, this genetic information cannot be read very easily by, for example, DNA polymerase, right? And so obviously genetic mutation will be introduced into daughter cells. All right, we're probably just not going to be able to go over one page in the textbook. Just look at that. This is just other functions of nucleotides. Just a couple slides. Look at those, and we're done. So what we have not discussed, let me give you a couple pages to read from the textbook, and that's specifically dealing with this DNA sequencing. Just hold on a second. DNA sequencing, we'll actually talk more about it in chapter 9, but there is some introduction to that in this chapter, and you had some sapling problems on DNA sequencing. Okay, so page 302. Let me go to the end. Read. It's actually a very, very cool process, and it's very easy to understand. So it's like PCR, polymerase chain reaction. Read pages 302. Through. Now there's a big box here talking about some applications of this sequencing. You can read it or not, but it's 2 pages. 310. 310. As I said, actually, there are two big boxes in here, so you can read the boxes. You know, you're not responsible for the boxes. It's just for your personal edification. But you should know what how the sequencing works and how it utilizes dideoxy nucleotides. Dideoxy nucleoside triphosphates. Right? How those are utilized in sequencing. Chemically, very cool process. All right, are there any questions about today?
Yeah, on one of the sapling questions, it said that, let's see exactly, it says this, it's talking about DNA and RNA. It says this contains the sugar ribose. So technically they both contain a ribose sugar, a 5-carbon sugar. Just one has a, is missing an OH, but it's still technically a ribose. And so I chose both DNA and RNA. And so I was wondering why. I understand, okay, it's probably just distinguishing between deoxy and just normal ribose, but isn't that technically, aren't they both still ribose?
Yeah, I mean, it's a matter of how the question is phrased and the context. Yeah, they're both ribose, but DNA is deoxyribose. Yeah. Yeah.
Okay, just wanted to know.
All right. I'll see you on Friday. Friday, today, I think yeah, you can start taking the test at 8:55 if you want. I think it's going to be available at 8:55, and you have 55 minutes until 10. So, all right. Let me know if you have any questions.
Thank you.
Bye-bye.
Thank you.